Good afternoon, good morning, or good evening, everybody, depending on where you are, and welcome to this first bioinformatics tutorial put on by the Bioinformatics Core. My name is Larsson Omberg, and I work at Sage Bionetworks, who's one of the participating centers in the Bioinformatics Core. And today, I'm going to be talking about the first in a series of tutorials, and we're going to be talking about how to access the PCBC data and also how to explore the data that is currently stored in Synapse. And we envision this series, a little bit of background about it first, as a series where we start out very basic about accessing data, but then go on to explore what kind of analysis you can do with bioinformatics. And we hope to be covering such topics as data exploration and quality control to see what kind of information is available in data that you might have, or data in the PCBC and how you correct for quality issues. We would like to cover differential expression. We would like to cover eventually network analysis and also pathway analysis. So looking at what signals are available in that, and how does that look as when you project it onto a pathway. And then throughout the whole series, we'll be covering data visualization as well, and we'll have specific sessions on covering data visualization. And even though today I'm going to be talking about expression analysis mostly and exploring that and fetching that data, it's important to note that the data that's available from the cell characterization core and what's available in the data portal is not only expression data, but also methylation data and microRNA expression data. And as we go through, we'll cover looking at all of these different data types. But for this first one, what we're going to talk about is the idea of accessing data and how to explore it. And to do that, we're going to move over to Synapse, which is the tool that we at Sage Bionetworks have developed for working collaboratively in consortia. And if you go to the Synapse webpage, you're greeted with basically a login screen with a little bit of information how you get started. But if you haven't already created an account, the first thing you'll need to do is register for Synapse. And it's relatively easy. You need to choose a username, input an email address, and then your name. And once you have done this, you'll end up receiving an email verification, which it's really important that you press the link on that is provided in that email. Otherwise, your registration will not be complete. And if your registration is not complete, you won't get a user account in Synapse. Once you've created an account, you can go to the login page of Synapse and log in. You can type in your username, password that you've chosen when you create your account, and once you sign in, you'll be greeted by your space in Synapse. And so, depending on how long you've been using and how much you've been using Synapse, depends on how much you'll have. So, for example, this is the way it looks for when I log in. So, I have my projects, which are the projects that I'm currently involved in. I have other people's projects and files, etc., that I found interested in, so I've added favorites to those. And then I also have teams that I'm involved in. So these are collaborations that I'm involved in. If you look for me, for example, you can see there's a lot of things about stem cells and then a lot of things related to cancer. Those are the two fields that I work in. Specifically, though, what we're interested in is the progenitor cell biology consortium. And when you come here, everything will be blank, and it's important that you get access to this project because this project is actually private and only visible to people who participate in the PCBC. And so what you'll have to do once you've created an account, you'll have to reach out to me and I will add you as a participant in the team for the PCBC. And instructions for this is available on the main page for the PCBC. If you look at bioinformatics and genomics tools, you can click on Synapse and there's the actual information. And this is, I agree, a little bit clunky, and we're working on making this registration process a little simpler. But in the meanwhile, you'll have to do that, and once you have done that, you can go to the progenitor cell main page, and to make sure that it pops up in your main entry, you can click the little favorite button right there, and then once you log into Synapse, it will be in the front page for you and easier to find. And then what I'm going to do today is go over a little bit about the different parts that we provide. But before that, I'd like to highlight the fact that if you run into trouble and you need help, there's a help forum which is staffed by people both at Sage and then also people from the Bioinformatics Core if you're asking for specific questions on the PCBC, and you can ask questions that relate to that. So, for example, we can see what other questions have been asked by other people, and it turns out there's only one question currently that has been asked. But if we go back to the front page, if you have a question, what about this data? Let's assume, and I can continue. And of course, if this is a question that's already similar to something that might have been asked, we'll show you that you can look for those. But if it's really a new question, you can type in details about your question. And if you want to address it specifically to the PCBC, it helps if you choose to say that it's related to the PCBC. Then the question will be directly emailed to me and other participants, whilst if you do a general question, it will ask it to anyone who's participating in Synapse. It will eventually get answered, but it will get quicker to answer if you choose to use the PCBC. I'm going to close that out and go back to the main page of the PCBC. And basically what we have is, in the project, we have a wiki that describes the content, and then you can also explore all the files that have been contributed to the project. But it's generally easier just to use what we provided in the wiki. And we divided it up into just certain categories that might be useful: exploring your signal in the self-characterization data, downloading data, exploring how we process the data, and then analysis results that have come in. And then links out to other tools that might be useful, such as enrichment analysis from Cincinnati Children's Hospital. And I'm going to start at the top level, which is actually, you have a signal that you're interested in exploring, or you have a set of genes that you want to see how they behave in the people data, and provide a tool for that. And then I'll move on to more detail. I want to download the data and explore the data locally. So let's start by looking at exploring the signals. And so what we have provided is we have provided basically a viewer into the expression data. And so if I scroll up here a little bit, what this is, it's a hierarchal clustering that's being done on all of the expression data that's currently in Synapse. So when data is added, this will be updated as well. And the way it works is that you can input the set of genes that you're interested in exploring, or you can choose specific pathways that you're interested in. So currently, it's using a list of genes, and it's displaying those genes along here, and then all the samples across here. And they're highlighted currently by the differentiation state. So definite embryonic body, ectoderm, mesoderm, others, which are actually tissues, and stem cells. And you can see that stem cells cluster by themselves in the different differentiation states differentiate across. And that's because the list of genes here are genes that are very relevant for the differentiation states that were chosen. We can also choose to look at specific pathways. So we can choose a KEGG pathway, and if we go down to the bottom, we'll choose the Wnt pathway. What it's doing now is pulling out all those genes and the expressions that are part of the Wnt pathway and showing how those cluster across the samples. Or we have also pre-computed all the differentiations between different states or cell of origin, and also differentiation based on state, and we can choose those gene lists. So, for example, we can choose those that are both in ectoderm and those that are derived from blastocysts, human embryonic stem cells, versus those derived from amniotic fluid. And you can then also further filter those down by specific pathways. So this gene list is enriched for the following PEG pathways. And if I click on a specific pathway, it filters down to those genes that are part of that pathway and also enriched, for example. Let's go back to the gene list, which is, if you're coming at this where you have a data set, you've already found a set of genes that are interesting, you want to see how those genes behave in this data set. So we can replace these, for example, and we can just pick a few genes that SOX2, MYC, and KLF4, and maybe NANOG. So we're picking 4 genes that are used to differentiate stem cells, for example, and we can see how those genes now behave across all of the samples. This is a lot of samples. We might not want to see all the different differentiation states. So what we'll do is we'll filter them down and only look at those cells that are part of the embryo body and the stem cell state, basically. So undifferentiated and those that are slightly differentiated. And now we basically see a new clustering, and we can see that basically the EBs cluster pretty well together with the stem cells, and we can see how the expression changes across the samples. Now you see this bright outlier here, so this might be a quality issue where one of the samples is measuring the MYC, for example. And then it's important that you can also change what you're coloring by. So, for example, we can add gender. So this now will tell you what the sample of the sample where they're derived from. And you can see that basically the gender distributes evenly. So this is not a signal that's driven by gender. We can choose line type. So that will tell us whether it's a human embryonic stem cell or an iPS derived stem cell. And we can also pick cell of origin. And so you can see basically this is very robust for the EB versus stem cell. And the important part though is I'm just picking something randomly out of the air basically, but the point is that you can take your set of genes and see how they behave in this. And once you've found something that could be interesting, you can explore it further. So you can basically see what's the gene, some more information about the genes. You can search within those that gene list as well. So if you do choose a path where the significant enriched gene list, you can actually find out what genes are in it. And then importantly, you can just download this data. So what this does is it fetches those, in this case, 4 genes and gets expression data across all the samples for these 4 genes. And then you can click on an enrichment analysis, which take these 4 genes and pass them over to the top gene program, where you can look at how well those genes are enriched for different classifications. So if we go back to the top here, you can also see where did we go to that heat map again. So where did this data come from? That's the important part. And so we have information here that this data is actually derived from something that's identified by a SYN ID, and this is how we file in Synapse. So we can actually click on that, and we can see where this data comes from. And this is all the expression data for all the samples that are part of the cell characterization core. And in Synapse, you have a little bit of information about it. You can see the size of the file. It has the ID. That's really important. We're going to use that later to actually fetch the data. And then it has information about the history, so we can see currently there's only one version of this file. And then you can see a little bit of what the file looks like. And then where were this file derived from? So what was done to achieve these expression calls? Well, for that we have a provenance graph, and if we expand, we can see that this is something that used concatenation, which uses script, and the files in this folder. So basically this file was used by combining the files in this folder. So we can actually go over to that folder and see what these files look like and what they contain. And what you'll notice, as it loads the page, it's a long list of each of the samples with their FPKM, which is a measurement of expression, both for genes and isoforms. So if we click on one of these files, again, we can download this file, so this would be the expression call for just one sample instead of the combined them all. But then again, and more importantly, we have the provenance of it. And so what this says is that this file was generated by running couplings. And what you can see there is an older version of couplings, so maybe it's time to rerun this data with the newest version of couplings. It used the BAM file as input, and it used the gene model that's available from Standard. And then you can see the script that was run, which tells you the parameters that was run to align or to call the expression knowledge using couplings. And then the BAM file, you can find out where that was derived from. Well, that was actually run using PopPat. Again, the parameters are available in the script, and that was used using the FASTQ file. So you can download this data at various stages of how raw you want it, depending on what kind of analysis you want to do. But in reality, the easiest is actually to go back to the original data. And so we'll go to the wiki, and we'll go to downloading. And so if you wanted, I showed you the file for the expression calls. Similarly, we have files for the microRNA and the methylation, and it shows the information about the version and who uploaded it, and a short little description. And then on top of that, we also have metadata and the raw sequencing data that I showed you as we track back in the provenance graph. So let's go back to this expression call data, and let's assume I want to download this for further analysis. I can click on it right there, and it starts downloading it. It's a 53 megabyte file. But most of the time when you're actually fetching data and want to analyze it, you actually might not want to just pull down the file. You actually might want to work with it directly in a programmatic way. And I know we're jumping very far here from interacting with it on the website to using a programmatic client. And so what I'm going to show here is using IPython, which is very similar to RStudio. It's a way of it to write programs and interact and do computation on it. And if that sounds advanced, think of it just as basically a super advanced calculator. I can type in a formula and it can execute that formula and make it a little bigger so it's easier to see. And what I'm going to do is I have imported some of the things that I will need. And what I'm going to do is I'm going to fetch the file that we talked about earlier. So I'm going to do syn get, and the ID of that thing was syn 224779. So that was the file we were currently looking at. And what this does is it goes to Synapse, fetches that file, and downloads it to the local computer. Now it's going to be very fast as I do this, and the reason it's relatively fast is because I've already fetched this file. So all it does is it goes to Synapse and asks, is there a newer version of this file? If there's not a newer version, use the one that I've already fetched. And then we can see if we look at this, this file has some information. We can see where it's stored. But more importantly, we probably actually want to read it in so we can do some analysis on it. And so I'm going to use a thing called pandas, and I'm going to read this in, and we're going to read the file that's stored in the path. And it's a tab delimited file. I know that because it ends in TSV. So I know the separator is equal to TSV. And then I also know that the gene names are stored in column 1. So I want to use that as an index of this. And I'm going to store this in a variable I call EXPR, short for expression. And so if I now run this, I'm now reading in that file into memory. And if I do, I can now look at what this file looks like by looking at the head of this file. And what you notice, this is exactly the same kind of information that we saw in the preview on the website. So basically, we can see that we have gene names along the rows, and then we have sample names along the columns. We also have some information where the Hugo ID as the name, and we also have the ensemble name and the position in the gene that are located, and then we have the expression values. And if we're interested about this, we can actually we can add information about this as well. So we can actually let's assume that we want to see what does this gene EPM1 look like. So we can extract that gene EPM1, and we can extract the expression value, and then we're going to plot a histogram of that. And what we see here is that we end up with a histogram, and this is the expression value of those genes across that. So this is the kind of thing that we'll be doing in more advanced sessions. We'll be processing the data and looking how the genes look. And we can do a comparative analysis, very simple here. We can actually look at this, and then let's compare this to NANOG, like that. And I just need to transpose this to make the plot work properly. And if I do this, I now can see how the distribution of the expression of two different genes. And so this is I've fetched the data, I've looked at specific rows in the data, and I can even move further than that. I could do, if we look at this matrix, it's actually very large. It's about 57,000 different probes, and that's a lot. So let's filter it down to something more manageable. I'm going to do the standard deviation of each gene. I'm only going to keep those that are above 10. So I'm keeping the most variable genes in this. And then what I can do is I can extract those, and I'm only going to extract the expression values for those, and I'm going to store that in the variable small. And then we'll just look at how many samples we're left with when we do that. And the reason I'm decreasing it is just so it's a little bit more convenient to compute on. And so now we have something that's about 7,000 genes versus 170 different samples. And now I can actually do an STD analysis, which is basically a PCA analysis on it. And so I'm going to now look at the small variables and see. And this is a script that I have that very quickly does a PCA to quickly visualize what the data looks like. So we're getting in a little bit about looking for quality in the data, etc. And we'll do more of this in future analysis. So this is running. It's taking a little while because it's running on my laptop. And what we see here is it generates a bunch of figures about the quality of data. So this is basically the TCA in summary form, and then the more traditional PC plot, so we can see what the distribution of the data is. And this would be a lot more interesting if we color-coded these by some information, such as what maybe the tissue of origin or the differentiation method, etc. But this is just showing how easy it is to fetch the data and then do a quick analysis to look at quality control, and then equally well we could have done something for looking for differential expression. So if we go back, I'm going to go back to the Synapse webpage just to summarize. We go to the front page. What we're providing here is we can provide a way for quickly exploring the expression data. We'll be soon going to be adding the same kind of exploration using the heatmap viewer for microRNA and methylation data as well. And then we allow you to download the data. You can just download it from the website, or you can interact with it directly from a programmatic client. And then this is kind of the things that we'll show for in future bioinformatics tutorials.